Crack open a chrysalis mid-metamorphosis, and you won't find a caterpillar changing shape. You'll find liquid, yellowish, cellular, the remnant of a living creature. The caterpillar dissolved itself, almost completely, using its own enzymes. And something even stranger, the butterfly that emerges from that soup still carries a memory the caterpillar formed. Most of us picture metamorphosis as the caterpillar quietly rearranging its parts inside a protective shell, a slow renovation. That image is wrong. What actually happens inside a chrysalis is one of the most structurally extreme events in all of animal biology, and it raises questions scientists are still actively debating. The chrysalis itself is not a silk cocoon. That is what moths spin from external silk threads. A butterfly sheds its outer skin one final time in a process called pupation, and that shed skin hardens into the chrysalis shell. The caterpillar has made its own container out of its own body, and then the destruction begins immediately. Inside, the caterpillar releases proteolytic enzymes, chemicals that digest its own tissues from within. This is histolysis, from the Greek for tissue and loosening. Within hours, muscles, fat tissue, and digestive organs liquefy into a nutrient-rich cellular fluid. Even large portions of the nervous system are broken apart in the process. But certain structures survive the dissolution. Hidden inside the caterpillar since it first hatched from the egg are compact clusters of undifferentiated cells called imaginal discs. They sit dormant through the entire larval stage, chemically suppressed, protected. When histolysis sweeps through the body, they are among the last things left intact. Each imaginal disc carries the blueprint for a specific adult body part. One pair will become the wings. Others encode compound eyes, antennae, legs, and the proboscis, that long-coiled feeding tube butterflies use to drink nectar. A single caterpillar contains more than a dozen of these dormant discs, each one a silenced instruction waiting for permission to run. The signal that unlocks everything is a hormone called ecdysone. As the caterpillar enters its final larval stage, ecdysone levels surge dramatically. This hormone overrides the immune suppression holding the imaginal discs dormant and triggers the caterpillar to seal itself inside a chrysalis. The caterpillar's fate was written on the first day it was alive. Here is the part that surprises most biologists when they first encounter it. Throughout the caterpillar's entire larval life, its own immune system was treating the imaginal discs as foreign tissue, rejecting them, suppressing their growth, like a transplant the body refused to accept. Only the ecdysone surge forces the immune system to stand down. The butterfly spent weeks being rejected from the inside. Once suppression lifts, the discs activate and begin feeding on the cellular soup the caterpillar left behind. This reconstruction phase is called histogenesis. The discs divide, differentiate, and self-organize into structures that look nothing like anything in the caterpillar. Wing scales, compound lenses, flight muscles, assembled from the raw nutrients of the dissolved body. The caterpillar does not become the butterfly. The caterpillar becomes the building material. For a monarch butterfly, the entire process takes between 8 and 15 days. In that window, a creature that spent weeks crawling and eating leaves is dismantled and rebuilt into an animal with a 10-centimeter wingspan, compound eyes containing thousands of individual lenses, and a brain rewired for long-distance navigation. The reconstruction is precise. The timeline is ruthless. The caterpillar and the butterfly share identical DNA, every chromosome, every gene, letter for letter. What differs is which portions of that code are switched on. The compound eye reads entirely different genetic instructions than the simple eye did. It is the same book reread with a completely different set of bookmarks, producing a completely different creature from identical source material. In 2008, a research team at Georgetown University set out to test something most biologists assumed was impossible. They wanted to know whether a memory, a specific learned behavior, could survive metamorphosis intact, not a genetic instinct passed down through breeding. A learned memory, formed by individual experience, the kind that should dissolve along with everything else. The team, led by neuroscientist Dr. Douglas Blackiston, worked with tobacco hornworm caterpillars, the larval stage of the hawkmoth Manduca sexta. They repeatedly exposed the caterpillars to a specific odor, ethyl acetate, paired with a mild electric shock. The caterpillars learned the association. They flinched, recoiled, and avoided the smell whenever they detected it. Then the caterpillars completed metamorphosis. Their nervous systems underwent the standard remodeling of holometabolic development. Most larval neurons are destroyed in the process. The adult hawkmoths that emerged had no reason, in principle, to remember anything a caterpillar had learned. Blackiston's team then exposed those adult moths to ethyl acetate for the first time as moths. A statistically significant portion of the moths still avoided the odor without being retrained. The findings were published in PLOS ONE with controls ruling out chemical residue carried physically through the pupa. A memory formed by one body had survived its near-total structural dissolution and resurfaced in an entirely different one. The result still surprises neuroscientists today. The leading explanation involves a structure called the mushroom body, a region of the insect brain associated with associative learning and memory. Research on Manduca sexta suggests that portions of the mushroom body persist through metamorphosis in a more intact form than surrounding neural tissue, potentially preserving the chemical trace of what was learned. It is a lifeboat in the dissolution. This forces a question with no clean answer. If a creature can dissolve the majority of its nervous system and still carry a learned association forward, then memory may be more chemically redundant, more physically distributed than neuroscience had assumed. The boundary between one organism and the next is suddenly less obvious than it looked. Holometabolism, this complete dissolution style of metamorphosis, evolved an estimated 310 to 350 million years ago during the Carboniferous period. Earth's forests were dominated by giant club mosses and tree ferns, and the first flying insects were diversifying into dozens of new lineages. The innovation appeared once, in a single ancestral group. It was never reversed. The leading evolutionary hypothesis is resource partitioning. A caterpillar eats leaves. A butterfly drinks nectar from flowers. They occupy entirely different ecological niches and never compete for the same food. One genome produces two non-competing body plans across a single lifetime, effectively doubling the species' ecological range without adding a single extra individual to the population. The payoff was extraordinary in scale. Insects that undergo complete metamorphosis, butterflies, moths, beetles, flies, bees, wasps, and ants, account for roughly 65% of all known animal species on Earth. Not 65% of insects, 65% of all named animal species. One innovation, evolved once in the Carboniferous, now underlies the majority of animal life on this planet. The chrysalis itself is more than a passive shell. It regulates gas exchange through microscopic pores called spiracles, maintains internal pressure, and in certain species can physically orient itself toward light. It is a functional life support system, sustaining the most extreme act of self-reconstruction in the animal kingdom while protecting the very discs doing the rebuilding from the dissolution happening around them. When the butterfly is ready, it releases an enzyme that softens the chrysalis wall from inside, then pushes through. Its wings emerge crumpled and soft, pumped full of fluid called hemolymph that hardens them over the next hour or two. What crawls out is structurally, neurologically, and behaviorally a different animal than what crawled in, not as metaphor, but as measurable biological fact. Not all metamorphosis is this extreme. A frog tadpole remodels dramatically but never dissolves its core structure. A grasshopper undergoes incomplete metamorphosis, adding body parts gradually across several molts, never liquefying. Only holometabolic insects take the approach of near-total deconstruction and reconstruction. It is the most radical version of metamorphosis in the animal kingdom, and by every measure, the most successful. Every year, monarch butterflies migrate up to 3,000 miles from Canada to a specific mountain forest in Michoacán, Mexico. The butterflies arriving in autumn have never been there before. Their parents were the last generation to make the return journey north. They navigate by the sun's position corrected for time of day and by an internal magnetic compass, running on instincts encoded in genes that survived unchanged through the soup. The caterpillar does not become the butterfly the way a child becomes an adult. It becomes the butterfly the way raw material becomes a building, through demolition, reconstruction, and something that looks almost like resurrection. Scientists are still mapping exactly what persists through the dissolution and what that implies for our understanding of memory and identity. What do you think survives the soup? Leave it in the comments.